Right now, cells inside your body may have already broken the one rule that keeps every living thing mortal. Normal human cells divide roughly 50 times, then stop. Cancer cells do not. They hijack your own biology to become something that should be impossible, immortal. This is not an invasion from the outside. It is a monster born from your own blueprint, and what nobody explains is exactly how it turns your body against itself. Inside every human cell, a silent countdown ticks away. Leonard Hayflick's experiments in 1961 revealed the hard truth. Normal cells can only divide around 50 times before they grind to a halt. This is no accident. The ends of our chromosomes, the telomeres, shrink with every split, like the burning fuse on a stick of dynamite. When telomeres run out, the cell enters senescence, a built-in shutdown sequence that prevents chaos. It is a safeguard, locking the door before damaged DNA can spiral out of control. But cancer does not play by these rules. It finds the loophole. In the 1990s, Elizabeth Blackburn and her colleagues uncovered telomerase, an enzyme that rebuilds those shrinking telomeres, patching the fuse so division never needs to stop. In most healthy tissues, telomerase is silent, active only in stem cells and embryos. In cancer, it roars back to life. Suddenly, the cell's expiration date is erased. This is how tumors achieve their unnatural immortality, by hacking the very system meant to prevent them. The Hayflick limit is supposed to keep us safe. Yet time and again, cancer finds a way around it. Why does this defense fail? Why can the monster inside us slip its leash, rewriting the rules of life and death? The answer remains one of biology's most unsettling mysteries. A mole on your skin, a polyp in your colon. These are not just harmless quirks of biology, they are the visible tip of an invisible iceberg. Every day, as your body churns out new cells, DNA copying errors slip through the cracks. Most are caught and repaired, but some slip by, leaving behind a permanent mark. Each mole, each benign growth is a clone, a tiny population of cells that has already started down the path toward cancer. They share the same first moves, a mutation, a burst of unchecked growth, a break from the discipline of the surrounding tissue. But most of these outgrowths never turn dangerous. Why? Because your immune system is always on patrol. Natural killer cells and T cells sweep through your tissues, sniffing out cells that look off, destroying threats before you ever notice. This is not a one-time battle. It is a ceaseless negotiation, a cold war inside your own body. Normal tissue is not a pristine landscape. It is a battlefield littered with the wreckage of failed rebellions. Even so, the numbers are sobering. Statistically, 1 in 4 people will face a cancer diagnosis in their lifetime. That is not fate. It is the sum of trillions of cellular gambles, most of them contained, a few breaking free. Cancer is not some alien force. It is the price of being alive, of letting cells divide, adapt, and sometimes betray us. Sunlight feels harmless, just warmth on the skin, a signal to wake up, go outside, live. Yet every hour under the sun is a molecular gamble. Ultraviolet rays slice through the outer defenses, striking DNA with enough force to snap chemical bonds. The result, cyclobutane pyrimidine dimers, tiny scars that twist the genetic code, sometimes missed by repair crews. Most of these wounds are patched, some are not. This is how a freckle becomes a warning label, a record of cellular Russian roulette. Step deeper into the body. The kitchen sizzle and backyard grill hide a subtler threat. When meat chars, amino acids, and creatine fuse into heterocyclic amines, compounds that, once eaten, are transformed in the liver into agents that attack DNA. Processed meats add another layer. Nitrites used to keep bacon pink and ham shelf stable react with stomach acids to form nitrosamines. These molecules slip into the nucleus, alkylating guanine bases and corrupting the blueprints of cell division. Each meal is a roll of the dice, the risk multiplied by habit, not by a single bite. But there is a king among carcinogens. Tobacco smoke delivers a chemical barrage, polycyclic aromatic hydrocarbons, nitrosamines, aromatic amines, and heavy metals. 79 known carcinogens in every inhalation. The damage is not local. It travels from lung to bloodstream, seeding mutations in organs never touched by smoke. Smoking alone accounts for nearly 1 in 5 cancer deaths worldwide, a toll unmatched by any other exposure. Cancer risk is not a single event. It is built from a lifetime of small, repeated insults. The question is not whether the damage happens, but how soon it can be found before the monster inside grows beyond control. Magnetic resonance imaging, or MRI, reveals hidden patterns in the body's soft tissues using a strong magnet and radio waves. It does not cut or touch, yet it can detect a tumor smaller than a pea buried deep in the brain or liver. The secret lies in how water molecules react to magnetism. Tumors, with their dense and disordered cells, trap water differently than healthy tissue. This creates a faint signal, a pale spot on a gray map. But even the sharpest MRI scan can miss a cluster of rogue cells if they are too small or too scattered to register. In 2015, researchers tried something unexpected. They trained pigeons to tell the difference between cancerous and healthy breast tissue slides. Given food rewards, the birds learned to peck at images of malignancy. After weeks of practice, their accuracy reached over 80%, matching novice radiologists. The point is not that pigeons will replace doctors, but that pattern recognition, whether by bird or human, can be surprisingly effective and sometimes even outwit technology. Artificial intelligence now scans millions of medical images, flagging suspicious spots in mammograms and MRI scans with impressive speed. The FDA has cleared the first AI-assisted pathology tool for clinical use, but every flagged result still needs a human eye. AI cannot read a patient's history or weigh subtle context. It can also trigger false alarms, leading to unnecessary biopsies, or overlook rare tumors that do not fit its learned patterns. The paradox is clear. The more we search, the more we find, but not every finding is dangerous. In prostate cancer screening, up to 60% of detected tumors may never cause harm, yet each one can spark a chain of tests and anxiety. Medicine must now decide what is truly worth seeing. Cancer treatment once meant unleashing chemical warfare on the body. Early chemotherapy was born from the aftermath of World War II, when nitrogen mustards, originally designed as poison gas, became the first weapons against leukemia. These drugs did not discriminate. They tore through every fast-dividing cell, hair, gut, bone marrow, and tumor alike. Survival sometimes hinged on who broke first, the cancer or the patient. By the late 1990s, a new idea took hold. What if medicine could strike cancer's unique weaknesses, sparing healthy tissue? Targeted therapies like imatinib zeroed in on mutant proteins, shutting down the signals that let cancer multiply unchecked. The results were dramatic for some, but cancer kept finding new escape routes, evolving past each blockade. Then came a leap that rewrote the rules. Scientists like James Allison and Tasuku Honjo discovered that cancer's greatest trick was hiding from the immune system, using molecular breaks to keep killer T cells in check. By releasing these breaks through drugs known as checkpoint inhibitors, doctors could unleash the body's own defenses. This was not just another attack, it was a recruitment drive, turning the immune system into a relentless hunter. In 2015, former President Jimmy Carter's metastatic melanoma vanished after treatment with pembrolizumab, a PD-1 checkpoint inhibitor. The world watched as a man in his 90s, once given months to live, went into remission. Carter's story is a testament to what is possible, not a universal cure. For some, the monster retreats, for others it adapts. But medicine is no longer fighting blind. The war on cancer is shifting from brute force to biological strategy, from collateral damage to outsmarting the enemy within. More cancer diagnoses fill the headlines, but the real story is written in the death rates. Since the early 1990s, cancer mortality has dropped by over a third, even as the number of new cases keeps rising. This is the measure that matters, not how many tumors we find, but how many lives are actually lost. Earlier detection, sharper treatments, and a deeper understanding of biology have shifted the odds. Behind the scenes, researchers are pushing into new territory with messenger RNA vaccines, custom-built instructions that train the immune system to hunt down cancer cells. These vaccines are not a miracle, not yet, but they point to a future where the immune system itself can be programmed to outsmart the monster within. The question now is not if cancer can be beaten, but how far human ingenuity can push biology before the balance tips for good. Cancer is not an alien curse, it is a glitch in our own code, a mirror held up to life's relentless drive. Today, cancer deaths are falling, but the battle is never abstract. Every advance is another reminder. Outsmarting this monster means outsmarting ourselves. What future do we dare to rewrite?